The image on the Shroud of Turin is not a painting, but a superficial discoloration of the outermost linen fibers, penetrating only 200 to 600 nanometers deep. This extreme superficiality means the color resides solely in the primary cell wall of the flax fibers. Crucially, the image is non-directional. It lacks the brushstrokes or capillary action expected from liquid application. When analyzed using a VP8 image analyzer in 1978, the shroud revealed inherent three-dimensional information, meaning the intensity of the image correlates inversely with the distance between the cloth and the body it wrapped. This unique property is impossible to replicate through standard artistic techniques like painting or scorching. The body image itself is a negative, while the bloodstains are positive, indicating two distinct formation events. This combination of ultra-thin coloration and encoded spatial data immediately challenged all known medieval forgery methods. The Shroud of Turin Research Project, STURP, conducted extensive non-destructive testing in 1978. X-ray fluorescence and microchemical tests confirmed the image areas contain no pigments, dyes, or stains commonly used in art, ruling out tempera, watercolor, or fresco techniques. Specifically, the image areas lack the iron oxide or vermilion expected in blood representations of the era. Instead, the coloration is chemically consistent with oxidized cellulose, suggesting a rapid chemical change in the linen itself. Ultraviolet photography revealed areas of water damage and scorches, distinct from the body image. The blood areas, however, tested positive for hemoglobin and serum albumin, confirming they are actual biological material, not pigment. These findings established that the image mechanism was fundamentally different from any known artistic or chemical process of the Middle Ages.
Scientific theories propose several mechanisms for the image formation, none universally accepted. The Maillard reaction hypothesis suggests that chemical compounds from a decomposing body, reacting with the linen's starch and moisture over time, cause the discoloration. However, this process is too slow and would not produce the observed superficiality. A more rapid theory involves a burst of energy, such as a corona discharge or ultraviolet radiation. A high-voltage electrical discharge could cause localized oxidation of the linen fibers, mimicking the image characteristics and the superficial depth. Experiments attempting to replicate the image using excimer lasers have successfully produced similar coloration depth and non-directional properties, suggesting a powerful, instantaneous energy event was involved in the image creation. Detailed analysis of the reddish stains confirmed the presence of human blood, specifically type AB. This blood type is relatively rare globally, but common in populations of the Middle East. Spectroscopic analysis indicated high levels of bilirubin, suggesting the individual suffered severe trauma, consistent with massive internal injuries and hemolysis before death. The flow patterns of the bloodstains are anatomically correct for a crucified man, showing distinct flows from the wrists, feet, and a large stain consistent with a spear wound in the side. Crucially, the bloodstains were applied before the body image was formed, as the image mechanism did not affect the chemical composition of the blood, only the surrounding linen. This sequence implies the blood was already dried onto the cloth when the mysterious image-forming event occurred.
Microscopic analysis conducted by Swiss criminologist Max Frey identified numerous pollen grains embedded in the shroud's fibers. Frey claimed to have found pollen from 58 plant species, 45 of which are native to the Middle East, particularly the region around Jerusalem. Specific species, such as Gundelia tourneforti, a thorny plant common in Israel and Jordan, were identified. The presence of these specific flora suggests the cloth spent time in the Near East before its documented appearance in Europe. Furthermore, imprints of flowers, including Zygophyllum dumosum and Cistus creticus, were allegedly found near the head area, suggesting the body was covered with local vegetation consistent with burial customs of the 1st century Judea. In 1988, 3 independent laboratories, Oxford, Zurich, and Arizona, conducted radiocarbon dating on small samples taken from the shroud. The results were highly consistent, placing the origin of the linen between 1260 and 1390 AD, corresponding precisely to the shroud's first documented appearance in Liry, France. This dating strongly suggests the cloth is a medieval artifact, contradicting the 1st century origin implied by the pollen and blood evidence. The C14 dating process involved cleaning the samples rigorously to remove contaminants before analysis. The combined statistical probability of the medieval date was calculated at 95% confidence. This result remains the single most cited piece of evidence against the shroud's authenticity. The 1988 radiocarbon dating faced significant scientific criticism regarding sample selection and potential contamination. Critics argue the tested sample was taken from a rewoven patch, known as the invisible mending or the raise patch, used to repair damage from a 1532 fire. Chemical analysis of fibers near the C14 sample area showed higher concentrations of foreign materials and dyes, consistent with medieval reweaving techniques. Furthermore, the presence of biological contamination, such as bacteria and fungi, accumulated over centuries, could introduce younger carbon, skewing the results toward a more recent date. A 2005 study suggested that the high concentration of carbon monoxide trapped within the fibers from the 1532 fire could also have artificially lowered the apparent age.
Chemical analysis of the linen fibers revealed an unusually high retention of vanillin, a byproduct of lignin degradation in flax. Vanillin typically degrades completely within 1,300 years under normal atmospheric conditions. The measurable presence of vanillin in the shroud fibers suggests an age closer to 1,300 years, aligning with the 1st century origin, rather than the 600 to 750 years indicated by the C14 test. This finding directly contradicts the radiocarbon results. Additionally, the linen itself is a high-quality 3-to-1 herringbone twill weave, a complex pattern that was rare but known in the Roman era, though it became more common in the Middle Ages. The VP8 image analyzer, originally designed for NASA to process planetary images, demonstrated the shroud's unique spatial data. When fed the shroud's image data, the analyzer translated image intensity into vertical relief, producing a perfect 3-dimensional representation of a human body. If the image were a painting or scorch, the resulting relief map would be distorted and meaningless, as intensity in art does not typically correlate linearly with distance. This 3D encoding remains one of the most challenging aspects for forgery theories to explain, requiring a mechanism that somehow recorded distance information onto the cloth. The data suggests the cloth was draped over a body that was not in contact with the cloth in all areas. The scientific evidence presents a profound paradox. The radiocarbon dating places the shroud firmly in the medieval period, consistent with its historical record starting in the 14th century. Conversely, the image chemistry, the superficiality of the coloration, the presence of type AB human blood, the vanillin retention data, and the encoded 3-dimensional information all point toward a unique, non-artistic formation event that defies known medieval technology and suggests a much older origin. The ongoing debate centers on whether the C14 sample was flawed due to contamination or reweaving, or whether the image formation mechanism itself is so unique that it requires a complete reevaluation of historical and scientific assumptions regarding this artifact.